I'm going to talk about characterizing biological tissues for the development of medical devices, regenerative therapies, diagnostics, and in vitro organ models. And if those things don't sound like they go together, it's because they don't really, and I've just put them all together, because that ties together my academic journey to date, which has been 5 years, as Edmund said, here at the University of Limerick, 3 years then out in the wilderness at RCSI and University of Pittsburgh on a Marie Curie Fellowship, and then 2 years BBSRC Fellowship at Imperial College London. So, at the risk of insulting people's intelligence, I'm going to start at the very base level and describe what a biological tissue is, because I know that there's quite a mixed audience. So atoms combine to form molecules, molecules interact to form cells, cells secrete and regulate their extracellular matrix, and these cells and extracellular matrix combine to form a tissue. So the tissues then go on to form an organ, and the organs interact to form an organ tissue. So in this case, kidneys and bladder combining to drain urine from the body, and the tissues of the bladder combining to form the bladder organ. But there are four types of tissues. There's epithelia, which cover and expose surfaces. They line internal passageways. There's connective tissue, which I'm going to be dealing with today, which fill the internal spaces between cells and tissues and store energy. Muscle tissues, which can contract to produce movement, and neural tissues, which conduct electrical impulses and carry information. So looking then specifically at connective tissues, there's the ones that people will probably be familiar with. There's supportive connective tissues, which are cartilage and bone. And then there's some more probably unfamiliar ones, or you mightn't have known that they were connective tissues. There's blood and lymph. And then the ones that I'll focus on primarily today are connective tissue proper, which loose and densely packed fibers, such as you'd find in the arteries or the urethra. And this tissue is composed of collagen fibers, which give the tissue its strength, elastin, which gives it its ability to recoil and store and dissipate energy, and then the ground substance, which is a gel-like substance which contains a lot of secreted factors in the cells themselves. So I did my PhD here. I know there was no need for the map, but for consistency I included it. We all know where we are today. I did my PhD in the MSSI 1. This is MSSI 2, MSSI 1. Looked lovely, but not as lovely as this. And my PhD was funded under the BMERM, the short-lived BMERM, Biomedical Engineering and Regenerative Medicine, under the supervision of Professor Michael Walsh, who was a PI on this programme at the University of Limerick. And I should say also that I spent some time at McGill University during my PhD under the supervision of Professor Razer Mongrain. And really the focus of my PhD studies was to employ different characterization techniques to distinguish different tissue properties. So the main ones that I went through were mechanical testing, where you deliberately deform the tissue in order to generate a stress-strain curve. I know stress-strain can be worrying terms for non-engineers, but basically it's just if you deform a tissue, how much force does it require to do that per unit area? And from these curves, you can discern like the modulus, or its resistance to deformation, its strength, how much stress it can absorb before it fails, and toughness, how much energy it can absorb before it fails. And then there was composition, looking at the composition of tissue using spectroscopic laser techniques, so deliberately shining lasers on the molecules within the tissue and seeing how that light scatters, is reflected, or is transmitted. And this gives you what's called a spectra. And from this collection of peaks here, you can discern how much collagen, lipid, or calcium is in that tissue. And then finally, there was morphological characterization, which is just imaging using primarily electron microscopy and computer tomography. And this was just to look at the different morphology, the content, the location of the tissue. So that's kind of just to go over what a biological tissue is, the different characterization techniques that I would have used during my PhD, and now onto some of the findings. So the tissues that we focused on during my PhD were from diseased tissue from the carotid artery, from the aorta, aortic aneurysm, swelling of the aorta, and atherosclerotic femoral arteries. And the reason we focused on these particular arteries and tissues is because this is what we could get. So we would drive out to the hospital on the day of surgery with a little bucket, and our bucket would be filled with these various types of tissues, and we would take them back. And the aim of that procedure, or that act, was to characterize the tissue to better inform medical device design. So in the case of the carotid and femoral artery, to better inform the design of stents and catheter balloons, which are intended to interact with these diseased tissues, compress them to restore blood flow, or in the case of aortic aneurysm, to better predict when an aneurysm might rupture based on how the composition, morphology, and mechanics interact. So dealing first with the femoral arteries, you can see here that we generated spectra using spectroscopic techniques. We were able to see the different degrees of lipid, collagen, and calcification in there. Calcification shouldn't really be in your arteries. This is more common for bones and dental tissues, but due to the disease process, calcification can build up in these arteries. And what we were able to do then was to relate the amount of calcification or the amount of lipid and collagen in the tissue to the mechanical properties, in this case toughness, so how much energy it can absorb before it fails. And we use this then to predict the tissue's response to stenting and inform medical device design. So similarly here, for the carotid vessels, we use CT, computed tomography, where you would use a lot of X-rays to develop kind of a 3D rendering of what it looks like in terms of density. And you can see here the dark grey to black indicates areas of calcification. So we were able to relate the formation, the density of the calcification to the actual mechanical properties, how it would fail, and again using this to predict tissue response to stenting. So again, trying to inform medical device design. Moving on to abdominal aortic aneurysm, we were using SEM, scanning electron microscopy, and you can see calcifications here in green. And we were relating the presence of calcification to the mechanics again, how it restricts the movement and of the tissue and the deformation of the tissue, and using this to predict the location of aneurysm and rupture. So this was all very interesting, but in the case of my project, it didn't actually inform any medical device design, or it didn't actually influence any diagnostics, which is why the next part of my academic journey was so interesting, and it was actually using the characterization of biological tissues to successfully develop a medical device and to commercialize that medical device and actually deliver it to the clinic. So urinary catheterization is a very common medical procedure where you insert a catheter through the urethra into the bladder in order to drain the urine from the bladder and prevent leakage during an inpatient stay, say if you were having a procedure and you needed to be given a general anesthetic. But it has this anchoring balloon at the end, and the anchoring balloon is intended to anchor it in the bladder and drain the urine so that you can't accidentally pull it out. Now, it's a blind procedure, and I'm going to try and play these videos here. It's a blind procedure, and what is intended to happen is as follows. The catheter is inserted through the urethra into the bladder where it can drain the urine, and the anchoring balloon is inflated. So that's what happens 99% of the time. But in a small amount of cases, what will happen is the catheter will be inserted, the clinician will believe themselves to be in the urethra, and it will be inflated in the tissue, causing an injury and resulting in potentially stricture disease, potentially losing the use of your urethra. So what we wanted to do was we wanted to develop a medical device to prevent this type of injury, and we were going to do this through tissue characterization. So here is the medical device. Sorry if people can't see my pointer. I'll get it up again here. So this is the actual device here. This is a lovely marketing picture for it. It's called the Transurethral Catheter Safety Valve, the TUX-V, and our spin-out company that was designed to commercialize this was Class Medical. But here you see the device. It's just basically a pressure release valve with the flow restrictor, and the intention here is that the pressure release valve will trigger at the point before tissue injury can occur. So you'll see it in cross section here. Here is the valve, here is the restriction. But at what point do we want, at what pressure do we want the valve to actuate to prevent damage? So what we did was we obtained porcine urethras from abattoirs, we obtained human urethras from gender reassignment surgeries in Germany, and we put them in this experimental setup here where you can restore them to their in vivo length. You can keep them at 37 degrees in 100% humidity, and we intentionally inflated a urinary catheter inside there. We monitored how the tissue reacted, and we recorded the pressure. And what we were looking to do was intentionally find the point at which damage occurs and then limit that damage. So I'm going to play you a video here, if I can come across. There we go. And what you're looking at here is a catheter being intentionally inflated on the right-hand side there of that human urethra sample. And you can see as the pressure increases in the anchoring balloon, the tissue deforms until the point that it can no longer deform and it fails. And this is the type of injury that people were experiencing because of sorry, I'm just looking for my pointer again here. Here we go. And this is the type of injury that we were hoping to avoid. So what we did was we related catheter balloon inflation pressure to internal diameter, and we found that reliably above or under 100 kilopascals of inflation pressure, damage did not occur. And whereas above this, rupture could be seen. So we now had a value that we could set this device at in order to avoid injury. So we had this device manufactured and we used it in cadavers first in RCSI, and you can see here, using the standard procedure, clinicians were able to inflate the balloon up to above 500 kilopascals in the urethra, whereas using our device, the pressure was limited to 150 kilopascals, indicating that damage would have been avoided. So we subsequently used this in 100 patients in a functional study. The valve was actuated 7 times, indicating that 7 injuries were avoided. And this went on to become, after my involvement in this company, so this is what I was involved in, and then afterwards, under the very wise guidance of Michael Walsh, it was the first Irish company to be CE marked under the new medical device regulations. It's been used in over 4,000 patients, and it's currently being sold in Ireland and the UK. So that was kind of an exciting success story of characterising biological tissues for medical device design. So after that, I received a Marie Curie Fellowship, and I was kind of pivoting my expertise from tissue characterisation to tissue regeneration, and I was doing this initially in RCSI in Dublin under the supervision of Professor Fergal O'Brien. And the idea behind tissue regeneration is that instead of using a medical device to try and avoid or treat an injury, you're trying to use regenerative therapies to regrow tissue that is absent or damaged or needs to be replaced. And how you do this is you take a biomaterial or a scaffold. It can be a hydrogel, a scaffold, or a decellularized organ. You're combining it with some kind of regulatory signals, and then you're expecting the cells of the host to come in and repopulate that scaffold and remodel it. So here's actually what the process looks like. You have your biomaterial. Once it's implanted, proteins will absorb on the surface, and then you'll have immune cells infiltrate. These will secrete factors that allow native progenitor and stem cells to come in. They will start laying down new matrix, the collagen and elastin that I spoke of previously, and then finally revascularization will occur. So this is what regenerative therapies hope to achieve, and one of the key things in this is having an appropriate biomaterial or scaffold, and this was kind of the focus initially of my Marie Curie Fellowship. So what we wanted to do was we wanted to develop a scaffold that was based on the native tissue, so a mimetic scaffold that would function as a tissue until the tissue could be regrown. And what we did was we targeted the urethra. We wanted to be able to regrow the urethra for people who had actually already received an injury and needed new tissue. So we set about mechanically characterising it using physiological pressure profiles, looking at the different regional properties so that we knew do different regions of the tissue need different types of scaffold and regeneration, and then looking at the morphology and composition of the tissue. So looking at the elastin fibres here are the dark fibres, and collagen fibres here are the blue fibres. So we use this information from characterising the biological tissue to inform the design of a new tissue engineered scaffold. We're using collagen and elastin in the same ratios that occurred in vivo, and then doing a bi-layered approach. So here you have a dense desiccated layer, and this was to prevent urine infiltration, and then a freeze-dried porous layer. So SEM images here showing the dense inner layer and the porous outer layer intended to allow cells to infiltrate and remodel the scaffold. And what we did then was we compared it against the existing gold standards. So anyone who's familiar with tissue engineering will be familiar with seeing these 3 letters next to each other a lot. This stands for urinary bladder matrix, and this stands for small intestinal submucosa, and these have been used clinically as regenerative therapies. And what happens is you would take these porcine tissues, you would remove the cells, and you'd be left with just this extracellular matrix. And no one had kind of examined to see if this is an appropriate substitute for urethra, and can it act as an appropriate scaffold. And what we did was we compared our new scaffold in green here with the existing tissue, the blue, and found that the SIS and UBM were orders of magnitude more stiff, and their mechanical properties were very, very different, indicating that they wouldn't perform their function in vivo. And also looking at the morphology, it's difficult to see here. I don't know if anyone can actually see it, but these SIS and UBM are formed by densely packed laminates, so not in any way emulating the native tissue, which is quite porous, macroporous, whereas this bilayered scaffold was a much better fit for that. So that's what I did when I was in RCSI in Dublin. But then on the same fellowship, I also went to the McGowan Institute for Regenerative Medicine in Pittsburgh under the supervision of David Vorp. And the focus here was to utilise appropriate in vitro and in vivo modelling to evaluate these regenerative therapies. And what I've shown here is kind of the scale of what you would do when you're developing a therapy. You would assess it using high throughput methods, so 2D monoculture, before assessing it in a more physiologically relevant model, such as a small animal model or a large animal model. And the Vorp lab was very good at using these models to assess regenerative therapy. So I'll just talk first about the actual regenerative therapy itself. It was actually back to arterial tissue. So when you have a blocked artery in your heart, what will happen is a bypass surgery if a stent or an angioplasty won't work. And what you're typically doing to bypass these is taking a saphenous vein from the leg or a radial artery from the arm. Now, a lot of patients won't have an appropriate vein or artery because the conditions of those can vary. Also, they might have previously harvested these for another purpose. So you want a tissue regeneration approach here using, as I said before, a biomaterial scaffold and regulatory signals, a therapeutic, in order to encourage the host cells to come in and remodel that. So instead of using a vein or an artery here, you would be using a scaffold and a therapy. And that scaffold and therapy would combine with the native cells in order to regrow a native-like vessel. So the scaffold we used was based on Bombyx mori silk cocoons. So you're taking the silk fibrin protein out of these cocoons, you're dissolving it, you're putting it into a cast in order to make it into the right shape. You're then freeze thawing it into a porous tubular silk scaffold and electrospinning it in order to make it more mechanically robust. So you'll see some SEM images here, different days of degradation. So you can see the inner porous layer here intended to allow cellular infiltration, and then the outer electrospun layer there that's making it more mechanically robust. So that was the scaffold, that was this biomaterial aspect, and then we needed kind of a therapeutic that's going to encourage the cells to come in, and when they do come in, it's going to encourage them to behave in the ways that we want them to behave, to secrete the proteins and the regulatory signals that recruit progenitor and stem cells. So the therapeutic we used was derived from stem cells, which are often used as a therapeutic, and they're called extracellular vesicles, and these are secreted by most cells in the body, and they're used as little packagings, signal packagings, and they contain protein and genetic materials that can be used to signal to other cells. But what's been found that they can actually replicate the effects of their parent cells. So instead of using a regenerative therapeutic stem cell, you would use one of these, or you would use these instead, and they would be a cell-free approach to regenerative therapeutics. So what I'm going to do here is I'm going to just play this. This is how we got them into the scaffold. So you would have a small rat-sized scaffold that you see there, and you're using a vacuum infusion approach. So you're drawing the liquid out, which contains the extracellular vesicles, and they're becoming trapped in the then in the scaffold. So I'm going to go back now and get my pointer and move forward. So we had decided on a regenerative therapy that we wanted to examine, and now we needed to have an in vitro and an in vivo model to assess it and to evaluate this regenerative therapy. So first we looked at an in vitro model because it's higher throughput, it's less physiologically relevant, but there's less ethical issues surrounding a 3D scaffold culture. So what we did was we had a fibrin hydrogel. Into this we placed the arterial cells that the scaffold and the therapy were likely to come in contact with, and we exposed these to different concentrations of our therapeutics, so therapeutic here in orange, controls here in blue, and we were getting a very good increase in the secretion of collagen and elastin. So that's kind of the goal here, is that you want your cells to lay down new matrix so that you're getting a new tissue. So after we confirmed this in an in vitro model, we moved into an in vivo model. So it was actually a rat model, and we had some very good microsurgeons there, and they let me help out until I wasn't very good at it, and then they stopped me doing it. But here you can see an explant of what it looks like after you take it out. So new tissue grown here, and what we saw was that compared to the controls, yes, we were seeing a significantly increased level of collagen and elastin being deposited, and we were also seeing a proper expression from the cells that infiltrated it, so calponin for the smooth muscle cells, von Willebrand factor for the endothelial cells, and we weren't seeing too many macrophages in there. So this was deemed a success in an in vitro model, in a small in vivo model. So we then took it on to a large animal model. And so the trouble with transitioning from a small animal model to a large animal model is the trouble of scale. So now you're trying to make much bigger scaffolds, you're trying to produce much more therapeutics, but you're also having to change some of your technology as well. So again, I'm going to try and play these videos here. So this is what we ended up having. We had to scale the technology. It's still under a vacuum. You're still infusing your cells and your vesicles into it like this. But now, because it's so much longer, you're having to actually, almost like a spray painter, spray the cells along the inside and draw them out into it to ensure that you're getting good coverage the whole way along. So that was one of the issues of scale that we had. But we were able to move forward with that, and we did implant it. It was actually into sheep as a carotid interposition graft, and you can see here the graft after 10 weeks in vivo compared to the native carotid. And what we did see was that you were getting appropriate cellular infiltration, you were seeing proper deposition of matrix proteins, and that there was actually a difference in the mechanical properties here. So that's what I was doing in Pittsburgh. That's what I want to draw your attention to, different in vitro and in vivo models. How would you utilize them to develop and evaluate new regenerative therapies? So following on to that, I was awarded a BBSRC fellowship to go to Imperial College London and work in the lab of Professor Molly Stevens. And this was, again, something different. So initially tissue characterization, then tissue regeneration, and now diagnostics, even further away from where I started. But it was to do with breast cancer. So in breast cancer, you will have these cancerous growths growing within the breast ducts. These cancerous cells can metastasize, but also what they can do is they secrete their own extracellular vesicles. So like I said before, these extracellular vesicles, small little packages that cells release in order to communicate with distant cells and tissues. And it's believed that in the case of cancer, that these extracellular vesicles harbour actual cancerous material and can spread and metastasize, or at least lay the groundwork for metastasis. But also what they can be used as is diagnostic markers, and that's what we wanted to do. We wanted to investigate, is there significant difference in composition between cancerous and non-cancerous vesicles, and can they be used as diagnostic markers? Now, how do we do this? We used, well, initially we did it in vitro. We took these cancerous and non-cancerous cell lines, we cultured them, we gathered the media, we purified out the vesicles, you can see one here, and then we used high throughput automated Raman spectroscopy to characterise hundreds and thousands of these vesicles. Now, these are only nanometers on scale, but you can use automated trapping to trap them, and then automated Raman spectroscopy to generate spectra that characterise them. So because it's automated and high throughput, we were able to do this at very high numbers, so 14,000 extracellular vesicles in total. And what you get from each of these analyses is a spectra, like I said before, and this spectra is characteristic of the composition of these vesicles. So you have lipids, you have proteins, nucleic acids, glycans, and lipoproteins. And the relative height of these peaks and the areas of these peaks will give you an idea of how much of that is in there. So if when we compared all of the different cell lines, so the first two in green here, the non-cancerous, and the remainder are cancerous ones, you can see qualitative differences between the average spectra. Now, it's very hard to take this information and to process it and to use it as a diagnostic or even to see if there's differences. So what we did was called multivariate analysis, and this is PLSDA modeling. So you're reducing all of this variance into 3 different latent variables, which are far easier to compare. So instead of comparing large swathes of data that are all different spectra and very, very hard to compare, you're just reducing those to 3 variables that can be plotted on a graph like this. And you can see cancerous vesicles, cancerous spectra, cancerous characteristics here in green, or non-cancerous in green, and then cancerous in pink, seeing a good delineation, although there's some overlap. And we're all familiar with sensitivity and specificity. You'll have to take my word for it, it's pretty high. It's 97%. I don't know if people online can see what's behind this little window here, but we can't here. So yeah, we were able to predict cancer versus non-cancer at an accuracy of 97%, and this was validated by n equals to 2, so big N equals to 2, repeating the whole experiment again and seeing similar values. So that was kind of where my 10 years of research had led me to, and I wanted to become an independent researcher, as everyone kind of does at one point. And what I did was I kind of started examining the skill sets that I had generated over that last 10 years. So regenerative medicine, tissue acquisition, cancer diagnostics, therapeutic evaluation, very broad, very hard to see the link between them. And I was doing some brainstorming to write an ERC, and I'm going to just focus on the ERC. I wrote a lot of grants, and ERC was the only one I got. So I won't talk about the ones I didn't get. But, you know, brainstorming for these ideas, this is the hardest part of it. You're having to read so much, you're having to decide what you're going to spend the next 5 years looking at. And I wanted to ask an important question, and I wanted to also be able to convince others that I could do the work. So one of the first things I came up with was I'd use regenerative medicine to restore coral reefs. And this is before I did any reading on coral reefs or anything, but I thought it was a very noble problem that I could maybe apply myself to. But of course, there's people far more qualified than I am with more appropriate skill sets to tackle this important question. So then I thought maybe I could use tailored hydrogels to control the drug delivery of different therapeutics. And while this is probably an important question, I didn't have the right skill set to ask a very important question in this field. So I kind of had to go back to the drawing board and decide, how can I best apply myself and contribute to the scientific field? So what I'll show you next is the ERC idea and what I presented in my interview as slide 1. And then I'll kind of go on and show how my skill set kind of fits that and how it fed into that. So the story here is that as a species, we are facing into a global fertility crisis, whereby falling fertility rates will result in this kind of a curve instead of this kind of a curve. So there'll be far more elderly people versus young people. And it's anticipated that this is going to curtail economic growth and stall preventative actions against other pressing global issues. So in order to address this, we should be trying to preserve our ability to reproduce, but we're currently falling well short of that because male sperm concentration has been in decline for the last 40 years, decreased by 52% over the last 40 years, and is projected to go below the fertility threshold showing here in the middle of this century. Now, that is a projection that I use to my advantage because it's quite scary and catches people's attention. But the main form of male infertility is actually idiopathic, so nobody really knows why it's happening. There is other ways. There's illness, there's injury, there's obstructive, but the main cause is idiopathic, so unknown. And the treatment for idiopathic male infertility is empirical medical therapies. Now, empirical just means educated guesswork. So there's no mechanistic understanding of how the therapy works, and there's no way of knowing or evaluating how it is working. And like the models that exist to do this, to interrogate the mechanisms underlying empirical treatment strategies, they rely on culturing tissues from neonatal mice, which is unrepresentative of the human condition. And there's persistent low throughput methods, manual analysis that the current models employ that reduce their reliability and reproducibility. And so the urological field really lacks an appropriate model to accurately predict treatment response. And this is where the remodel project comes in. And the aim here is to develop representative, reliable and reproducible models of the male reproductive system using human tissue properties, high throughput testing and automated analysis. And I was able to convince people that it would be a good idea to give me a lot of money to try this. But how did I kind of justify it? Because I was now asking an important question, but how was I showing that I was the one to be able to do it? How could my skills be applied in this regard? And so justifying my involvement in male urological and reproductive health, I have experience in urology, I have experience in models, clinical collaboration, and then when you're talking about representative human tissue properties, skills in tissue acquisition and characterization, when you want to make something reliable, you make it high throughput, so you've got high numbers to reliably get statistical information. So using material fabrication skills, cell isolation and cell culture, and then making things reproducible and having automated analysis. Again, I have experience in automated Raman and therapeutic evaluation. So this is kind of how I justified my involvement in this project. And the idea then, the overarching idea being to completely disrupt this channel here of going from high throughput to high physiologically relevance. Why can't we have both? Why can't it be high throughput and be more physiologically relevant? Because even if you are talking about in vivo models of rats and pigs, they in no way kind of replicate a diseased state in a human. Well, they certainly don't represent, they're poor representation for healthy human, let alone a diseased state. So that was kind of where the objective for this project came in and what I'm hoping to achieve over the next 5 years. So with that, I would like to make a lot of acknowledgements. University of Limerick, RCSI, University of Pittsburgh, Imperial College London. There are a lot of names here. There should be a lot more names here because this work was done with the help of a lot of people and I'm taking credit for it now, but it's certainly a collaborative effort between a lot of people, and also to acknowledge my very generous and trusting funders. So thank you very much, everyone.
That was a really good story, really well told. There's time for questions. And I can't see if there's questions on the chat. Katrina, maybe let me know.
It's very nice and very interesting. Something that is probably not related to your to the project that you've done. The idea of studying corals in Ireland doesn't seem. I mean, if you can bring corals to Ireland, it would be an achievement. As for your journey into changing from one field to the other, how was it for you to change, to study from one field to a different one? Because maybe there are a few students here that they think that they've started doing something and they have to study, they start studying carbon atoms, they can only do that and they cannot change. How did you find your next jobs and how did you find your
So just for anyone who is trying to transition their research expertise and to spread out, it was certainly about finding the right people first, the right person that you want to go and work for first. So being motivated to work with a certain person and using your existing network. So my supervisor Michael was very good in identifying people who would be good for me to work with. And then after you've done that, there is the challenge, of course, of getting into the lab, getting the job. If you can't show that you have the right expertise, you're probably going to have to write a grant to come in, and then you're trying to justify, you're always trying to accentuate the link between what you're doing currently and how you're going to do what you do next. And I suppose then once you've done that and you've established that and you've made the connection and you've gotten the job, it's about a lot of reading, because you're really going back to the drawing board then. And every time you go back to the drawing board, Google Scholar should be your first point of call, trying to read up on everything. And certainly I do have a lot of blind spots in terms of what I've shown today that I might not have if I'd stuck with the one field. But I do think it is possible if you're identifying the right people for the right reasons, and then you're doing due diligence with your reading and your background stuff, and then developing the skills after you've done that in the lab.
Sarah.
So great talk, really interesting. So really lovely to see the journey. I'm actually going to go back to your work. So you know the models you built for, say, the arterial tissue.
Yes.
Did you ever consider? Well, two questions about them really. How many different polymers or matrices did you try before you fixed on the Bombyx Mori?
Yeah.
And also, did you ever try, rather than using them as implants, did you ever try use them as in vitro testing models? You know, for testing stents and like when they, like if you implant smooth muscle cells or an endothelial layer on them, did you ever consider actually using them then as an organoid, such for in vitro testing?
Yeah, and I think just to go back to your first point, why did we pick silk? I think you utilize the expertise that you have. So when I went over to Pittsburgh initially, we were collaborating with William Wagner, and he was providing synthetic materials for our degradable porous scaffolds. But the process of making those scaffolds, the electrospinning process, was very dependent on humidity and time of year and stuff. So the quality of scaffolds would vary. And then a PhD student came over to the lab who worked in India, where they have endemic native silkworms that they were working with. So using their expertise to develop these, to use these materials that we had access to, because you want to perform a lot of experiments, you want to gather a lot of data, you want to interrogate your system as much as you can, and if you don't have a good supply of materials, it's very hard to do that. And that was the idea between switching from the PEUU, polyethylene urethane urea, to silk fibrin. So that was kind of how we landed on the silk. And in terms of could we have used this as an in vitro model, I do think we could have. And what we actually ended up using was a fibrin type hydrogel, and you're putting the cells in there.
And just one more follow-on question then. Did you ever look at the impact of flow in vitro in the system?
No, we never did. We never looked at the effect of flow in there, and it's something that we should have done, definitely. It was very much a static model. It was how the compromise between, again, throughput and physiological relevance, how quickly do you want to find these answers out, and how representative does it need to be. And certainly, if we had been looking at endothelial cells, I feel like we would have had to incorporate flow. But because we were looking at the smooth muscle cells that exist within the matrix, where flow is a little bit more limited, we just wanted to see matrix deposition within that. And I think, like, if we had been looking at, say, endothelial cells and the effect of this therapeutic on those, then that would have been a very important factor. And we did, like, when we were looking at these models, we wanted to know patency as well was a big thing. Will they stay patent? So that is probably something we should have checked in vitro first before we looked in vivo. But we use our in vivo model to look at that as well.
Luke, you have a question?
Yes, I do. Thank you very much, Owen. I could follow it at a distance. It's really that in the Bernal Institute now, also to welcome. I have a question. You mentioned modeling mostly from the perspective of experimental models. How do you believe that, or do you believe mathematical modeling can play a role here?
Yes, I definitely do. I definitely do believe that mathematical modelling can play a role here. And I remember during my PhD, there would have been kind of a divide in the lab between those that were doing experiments and those that were doing computational. And certainly, and we all used to joke about how everyone believes the experimentalist except the experimentalists themselves, and nobody believes the computationalist except the person who made the model. And I think, like, the reason that is because when you're building a model, right, you're trying to consider everything, and you can iteratively develop the model, and you can make it so much better day on day. Whereas when you perform an experiment, you're kind of fixing yourself in that moment with those circumstances. Yes, you can develop it onwards, but certainly I think you have more room and more scope for iterative improvement with modelling. And it's definitely not my area of expertise, and something that I would love to recruit in and have someone educate me as well as give me credit for. But it's certainly, I think something that is probably underutilized. And I know Ted Vaughan above in NUIG is doing a lot of work with that within in silico kind of modelling. So yeah, very exciting field. Very jealous of those who are able to do it, and would love to learn more.
Well, to follow on from that, have you come across many people, because you've been in lots of different places now, have you come across people who have actually bridged that divide? Because you generally have, like in chemistry, you have the theoreticians and the experimentalists, and you rarely have somebody who, it's not that common to have somebody who does both.
Yeah.
It happens.
Yeah, so
have you come across that?
I have a colleague of mine, his name is actually there, Sebastian Callens. He's a Belgian man who worked in the Netherlands and did his PhD there. And what he was doing was looking at the influence of surface topography on cells. So what he would do is he would develop very interesting surfaces using computational models and using all sorts of parameters to control incrementally how the surface appears and how the surface is. And then you can use 3D printing to actually print these surfaces and see how the cells interact there. So I think the divide might be narrowing, and just one idea, the divide might be narrowing because experimentalists are able to actually match the control that a computationalist has using these techniques like 3D printing, and certainly like using the nanoscribe, you can control on the nanoscale. So you're looking at a very high level of surface control, and that's one really good example, I think, of witnessing someone bridge the gap, and it's incredibly impressive to see computational lining up with the development of a computational model and experimental model and the lining up of results, and it's really nice story.
Hi, thank you so much for such a nice presentation. Regarding the data set that you acquired in Imperial for breast cancer diagnostics, have you tried using other statistical analysis to maybe stage the cancer, or is it only for cancer or not cancer? Yeah,
that is such a good question. So you'll see here we used 11 different cancer cell lines, and the reason that this panel was established was to look at the different kind of HER-negative, HER2 negative, so the different kind of receptor status, the different origin, is it ductal, is it mammary? So, and we were able to see, now it wasn't with the same level of specificity and sensitivity, but we were able to see certain differences. And one of my colleagues was very good at entropy minimization to look at the differences between these. So you're minimizing the entropy in order to actually reliably look at the compositional differences between the different cell lines, and you're able to show that, certain, like here you might see that these three might be ductal cell line, might be from a ductal cell line, and they have really high lipid peaks here. So we were able to show that there is compositionally a difference between cells originating from different cancer types. However, as a diagnostic tool, it probably wouldn't be sensitive enough. I think as a diagnostic tool, it's more just yes, no. But as a research tool, then of course it becomes very interesting, and this is kind of where you have to, like, I really understate how hard it is to get these extracellular vesicles. These are a nightmare to get, and when you get them, they're filthy. They're covered in other proteins, and the yield is really low, and they're probably all agglomerated and stuck together. So using them as a diagnostic tool is a little bit far-fetched using the current methods of isolation. But as a research tool, it's very interesting to see the differences and trying to explain, why the different the cell of origin would produce an extracellular vesicle that is different from, say, something that is a different receptor status, or a different tissue of origin, a different ductal versus mammary, maybe, or something like that.
Just another question regarding your coral proposition. Because you have expertise in scaffolds.
Yeah.
But you wanted to make hydrogels. Why not scaffolds that can act as a matrix for then the corals to grow from there?
Yes.
Just have like.
Yeah, that was my idea. I was gonna develop scaffolds, like, really hard scaffolds to help regenerate, and you might put these scaffolds in the ocean, and then they could be recellularized. Yeah, but the spores of other coral would infiltrate and grow, and there are labs full of tanks of coral that are doing this, and fair play to them. It's very cool stuff. So yeah, so I mean, I think the message there is don't get too excited with a good idea, because you really have to test them first. And certainly don't write anything before you before you test your idea. Don't try write half a grant and then check the idea. Check the idea and then write the grant.
Okay. Thanks.
Can I just ask, how do you measure the entropy change of a cell line?
Sorry?
You were talking there about the entropy change.
Yes.
How do you do that?
Oh, Evan put me on the spot here now. I can share the link with you because, to be honest, I never fully comprehended how it was being done, but it was all being done through modelling.
Yeah.
Yeah. It was incredibly impressive, and I think this is kind of something you find when you work in these big labs. So the lab at Imperial was very big, and you have a really good range of expertise. And so, whereas my job here was, to isolate these vesicles, to examine them using Raman spectroscopy, to produce these results. The ability to analyze these results is a science in itself, and being able to see Simon being able to actually use these techniques to confidently say things that we couldn't say from just looking at the data was incredible.
I'm actually just finishing teaching the first half of a course, and it's on second law of thermodynamics. So it'd be, if you have a link that I could check, it'd be great.
Oh, definitely.
It'd actually be a nice application to show.
Oh, fantastic. Maybe you could help explain it to me as well.
Okay, thanks very much. If there are no more questions.
One more, I think.
Oh, sorry.
For anyone here that's thinking of applying for an ERC in their future, what's the best advice you can give them? What's the biggest lesson you learned? Start early.
Start early. If you think it's too early, it's nearly too late. It's a massive amount of work. And as I said, like coming up with the idea, like I'd say most of my reading was unrelated to male infertility. Most of my reading was on other things, on on drug screening, on in vitro models in general. And like certainly the first two drafts I wrote had nothing to do with male infertility. It was more to do with just in vitro models. And you never know when you're going to really get to the final iteration of what it is. Now, if you have a good idea and it's born out of the work that you've done, fantastic. Develop it out as much as you can and show everyone why it's such a good idea and why you're the only person that can do it. But if you're like me and you're looking at a blank page and thinking, not that I want to change the world, I want to get a job, is my motivation here. I would like a job and I would like to do something interesting while I'm doing my job. So it becomes very challenging then, where do you even start? But I would encourage people to, what interests you, like the coral reef, what interests you. Is it feasible? No, it's not, but it was worth reading a few papers on it. But what interests you? What can you convince other people that you can achieve? Where does your skill set naturally lead you to next? And is there a very interesting field out there, like male infertility? I'm being deliberately alarmist by saying that all men are going to be infertile in the next 30 minutes. But a reviewer, that'll that's a good argument. That is, and there's, if you have a paper, references to back it up, acknowledge that it's a projection, acknowledge that it's a limitation in the study, and that it could be quite alarmist. But there's a field that you could apply yourself to and use your skill set to advance that field, and that's what I'm trying to do. And I'm sure there's people who've been studying male infertility all their lives are rolling their eyes right now at this absolute cowboy who's after getting an ERC. But I'm coming into this field to apply the skills that I have developed and take a new and somewhat fresh approach to it by using tissue characterization, using high throughput methods, using automated analysis to try and advance the field. And if I fail miserably, at least no one else has to bother trying that again. But if I do advance the field, great. And that's just do it in a nice, scientific, reliable way.
Okay, thanks very much. Could I just thank Eoghan again.